let me be clear with my very first sentiment. Allulose is not a healthy sugar. There is no such thing. Due to the inhibition of cellular utilization of glycolysis, possibly by its action of glucose transporter inhibition, there is less pyruvate that is catalyzed into acetyl coenzyme A, with downstream effects leading to a reduced production of glycolytically produced ATP. I present to you an individual, a starving African child. AMPK also inhibits acetyl coenzyme carboxylase, which inhibits fatty acid synthesis because of its downstream reduced production of malonyl coenzyme A, which then disinhibits CPT1, or carnitine palmitoyltransferase 1. Glucose, fructose, and allulose all stimulate GLP-1 release. Allulose is not unique in this. It is not nature's Ozempic. So allulose is not improving your health. Your body is just adapting to a poison that is blocking the supply of energy. A ketogenic diet of 20 grams of carbs is considered healthy, but a carnivore diet of 0 grams is the most healthy. Notice how I did not mention calories.
Last question for you as we wrap up here. Let's bring those together and talk about allulose and how somebody might want to use that therapeutically as a sweetener to help increase GLP-1.
I was just talking about this with someone recently because I think it's very easy to fall into a logical trap, which is like, interestingly enough, allulose does promote GLP-1 production. And there are now data showing that it probably has anti-obesogenic effects, at least in preclinical models where you, say, feed mice a Western diet and give them stevia versus allulose. This is out of the Bickman lab. Allulose appears to protect against obesity in these mice, which is very interesting. So one could jump to the conclusion of, we know GLP-1 causes weight loss. We know allulose promotes GLP-1 production. We know allulose can have an anti-obesogenic effect, at least in mice. So therefore, allulose increases GLP-1 to promote weight loss. But that logic is flawed on a few fronts. High level, this could be something that is true, and unrelated, where there are possible metabolic benefits, but they're not mediated via GLP-1, because actually the GLP-1 levels induced by allulose, probably, at least again in mouse models, a 4-fold increase in portal vein levels, which is nothing to shrug at, still isn't like Ozempic, which is super physiologic levels, right? So, and that stick around for a long time. So I'm not convinced the metabolic benefits of allulose are per se due to GLP-1, although there does appear to be a pretty interesting signal in the literature. And it has a lot of other effects as well.
Let me preemptively start by pre-warning you that I'm going to preface this video with a long and meandering monologue of personal opinions. So down below are appropriately named chapters. Click on the one that contains the relevant information about the intended subtopic that you wish to watch and rewatch as needed. However, context and the human element are important, and they are intertwined in the background of why I'm making this video. So, let me humbly introduce myself. I am Obsidian Lion, and I am none of the following: a scientist, professor, biochemist, physician, doctor, nutritionist, PhD candidate, or anything resembling a professional or educational connection to medicine, science, or otherwise. What I do is suffer from Dunning-Kruger. I am a primarily carnivorous content creator, and I'm just a regular guy, probably much like you, the viewer. In this video, I'm going to attempt to actually explain allulose in a deeper context of its function, effects, and metabolism. If you clicked on this video looking for an in-depth analysis of allulose, you're going to want to stick around, because I'm going to deep dive into my personal understanding of the mechanism of action and metabolism of allulose intake, and explicitly explain how it works in the body. Because elsewhere there are a plethora of people that fluff up this topic, and to an extent metabolic science, and waste minutes to hours of your time while explaining mostly nothing. But let me get real with you for a second. It doesn't actually matter if you click to this video. What matters is if you watch this video. So let me be clear. If your attention span has been decimated by the mentally corrosive short form format of videos out there nowadays, but you have just enough self-awareness of this fact, then you should probably close this video right now and enjoy the regurgitation of the same talking points perpetually presented to the public in other videos. However, if you're running on ketones, your mind is sharp, and your ass is flat on that chair, ready to learn from a long-ass video, then congratulations. Your IQ is probably most likely above 3 digits, bordering on 5, and you're going to end this video edified and educated, ready to think about allulose and all its effects with a more comprehensive understanding, with a healthy smattering of my own personal bias. Warning, it's negative. Before I begin, I have to make a statement. Calories aren't real. Let me say that again. Calories aren't real. Before you proceed any further in this video, you must accept this concept. It is an absolute prerequisite requirement. Calories are a completely irrelevant metric of measurement that has nearly to completely zero utility in the nutritional and metabolic processes that underlie human health, fitness, metabolism, body composition, weight, and the gaining and reduction thereof. Don't believe me? I want you to answer a couple of simple questions. Question 1. I ate 2,000 calories of an omnivorous meal. What was the macronutrient composition of the meal? The answer is you don't know and you can't know. The caloric total says nothing about the meal, and the calories can be comprised in a near infinite amount of ways of calculating out protein, fat, and carbohydrate ratios, especially if you're going by logically inconsistent calorie loads that measure into the decimals, one example of which is a novel 0.2 calorie epimer of fructose. Question 2. I ate a hamburger consisting of 1 pound or 453 grams of ground beef at a ratio of 80% protein and 20% fat, and a 53 gram breaded bun. Calculating out things like cellular water, what did I eat in terms of macronutrients? The answer is approximately 90 grams or more of protein, approximately 100 grams or more of fat, and approximately 30 grams or more of carbohydrate in the form of starch. So, you can be sure you approached your daily protein requirements. The fat and carbohydrate would be enzymatically digested and utilized in the appropriate metabolic processes, and the caloric sum of which would total around 0 fucks given. Question 3. Explain how a calorie counting regimen would consider daily micronutrient requirements that include such totally calorically relevant substances such as magnesium, potassium, sodium, vitamins A through D's nuts, vitamins B1 through 40,000, iron, folate, iodine, etc., etc., etc. Question 4. Here's a picture of the 3 macronutrients' chemical compositions. Biochemically, explain the process by which these are converted into calories. You can explain how carbohydrates are enzymatically cleaved and absorbed in the small intestine, while fats are emulsified by bile, broken down into their constituent parts, and then undergo either storage in adipose tissue or beta oxidation within the cell, resulting in ATP, H2O, and CO2. And that protein breaks down to peptides and amino acids, which are then reconstituted into cellular, enzymatic, and muscle structures. But the real question is that I want you to directly look at these chemical structures and explain exactly how proteins, fats, and carbohydrates are converted into calories, aka heat energy. Where and how does that take place in the body? But finally, to really tie in the relevance to this video about allulose, question 5. I want people who believe in calories to explain exactly how one simple, epic, epimeric change turns fructose from 4 calories into 0.2. Explain how flipping that switch results in a 3.8 caloric reduction. Calories are not relevant and are logically inconsistent. In the land of calories, the macronutrient mass is king. But the main material of this video is allulose. The reason I'm making this video is because I'm kind of unsatisfied at how the people who are promoting allulose seem to just every single time not go super in depth into it. It seems to me that there is just simply not enough widespread mainstream information, just widespread mainstream disinformation, which seems to result in there being a lot of lying by omission, bias through personal or commercial obligation, or the worst part, a complete misunderstanding. But I shouldn't throw stones. This video is a glass house, and my ultimate goal is to get some sort of actual metabolic explanation out onto YouTube so that it eventually adequately stimulates the conversation and information dissemination. In other words, the ultimate goal is to eventually expose the truth and stimulate metabolic discussion about allulose and its mechanism of action. It sort of doesn't matter if I'm partially or fully wrong, as long as in the future this provides a foundation for people to get it right. So, there are videos about people using allulose and ingesting various doses, even up to 50 grams, and scientists such as Ben Bickman and Richard Johnson will spend a relatively short amount of time giving short explanation bites about how it works. And it's pretty much been hammered in from their short explanations that the mechanism of action is AMPK activation, GLP-1 stimulation, and blockage of fructose metabolism and absorption. Now, this is kind of fine, but it wasn't until I actually learned exactly what these cellular metabolites do and how they function that I believe I can truly theorize about how allulose works. Learning about human metabolic pathways, homeostatic glucose regulation, endocrine response, and nutritional biochemistry, among other things, over months of watching videos is why I feel semi-confident in making this video. Why I'm making this video is because pretty much what you will see on YouTube is, oh, it activates AMPK and GLP-1. Oh, by the way, I'm a board member of RX Sugar, buy our proprietary overpriced chocolate bars. They reduce the effect of unhealthy carbs. And don't forget to eat your healthy carbs, but make sure to keep them below the amount where it gets unhealthy. But enough about doctors being doctors, aka pharmacological salesmen, and let's get into the allulose. Personally, I've been using allulose myself pretty much daily for about 9 months now, and allulose is definitely not a placebo effect. Although there is some sort of tolerance development to it, every single time I ingest allulose, I am absolutely sure that it is somewhat effective. However, please bear in mind that I almost never ingest allulose on its own. It's 97% of the time combined with a cup of coffee. So take my N=1 anecdote with the context that it is muddied by inflammatory polyphenols and caffeine. Alright, let's get started. First, let me preface with my personal bias towards allulose. Let me be clear with my very first sentiment. Allulose is not a healthy sugar. There is no such thing. Humans are tentatively considered facultative carnivores, meaning although we are capable of deriving nutrition and a certain form of longevity from consuming plants and their associated protein and carbohydrate makeup, historically and metabolically, we thrive on a diet that is primarily or exclusively comprised of animal meat. The dietary requirement for carbohydrate from plant sources in any form is not 1 single gram ever. Allulose, no matter what the perceived beneficial effects are, is not an exception to this. Now, carnivore does not mean zero carb, since there is a minute content of carbohydrate in meat, but more on that in the future. Okay, so let me put it this way. We now know, thanks to the dissemination of information from the works of scientists like Richard Johnson, Robert Lustig et al. About the glycating, oxidative, inflammatory, dysbiotic, and lipogenic effects of sugar from exogenous sources, aka dietary carbohydrates in the form of simple and complex saccharides such as glucose, fructose, sucrose, dextrose, maltodextrin, starches, and other oglio and polysaccharides, and whatever other forms they come in. It doesn't really matter. All of them break down into constituent forms of carbohydrate mass to be metabolized in the body. The main or most common results being glucose, fructose, or galactose, or maybe result in lipogenesis or fat generation, depending on other factors. Glucose is a primary energetic substrate, wherein the cells of your body take it up and break it down to generate ATP. Fructose, when ingested, is metabolized and converted into glucose, but that's only a small part of what it does and can do. Increased circulating levels of glucose and fructose are contraindicated towards human health in that the human body is shown to operate in an optimal condition at a minimal and stable circulating level generated by breaking down fat with the absence of exogenous dietary sources. Increased glycation of cells, oxidative damage and stress, inflammation, superphysiological glycogen, fat storage, decreased metabolic rate, and ATP production, all of these result from carb eating in comparison to your body's natural intended metabolic function, which would be the result of an exclusively carnivorous diet. Allulose, in its studied mechanisms of action, seems to operate and exude uniquely different metabolic action and effects, resulting in perceived lack of these stated negative effects. However, this does not mean that allulose is a saccharide that improves health. Let me get straight to the point. Your body, on a proper human diet of the protein and associated fats of animals, creates the optimal operational levels of all your relevant biomarkers. To state it clearly, the homeostatic level of glucose and other metabolic markers in your body produced by gluconeogenesis derived from lipolysis is the optimal state of health. Allulose is in conflict with this state in a similar manner to other saccharides if you think about it. Let me give you a hypothetical. You have an obese individual, chronically consuming carbohydrates, elevating his blood glucose and spiking his insulin. Allulose, with its metabolic effects, would reduce his insulin resistance, his fat stores, and his hunger. Wow, allulose, the perfect sugar. Let's put this in literally everything and replace sucrose and corn syrup. We've found the answer. Okay, let me give you another hypothetical. I present to you an individual, a starving African child. No food to eat, nearly no fat stores to generate energy, emaciated and nearly dead from a lack of nutrition. Now, allulose is a healthy sugar, right? It's healthy, right? Once again, it's a healthy sugar. Would you give this totally healthy sugar that prevents lipogenesis, increases metabolic rate, and reduces hunger to this starving human? Conversely, we don't want to give fructose to this guy. Fructose is evil. It causes humans to lower metabolism and generate fat. And as we know, lipogenesis is not good, or something along those lines. Remember, fructose is the bad sugar. High fructose corn syrup and cane sugar are Lucifer and Beelzebub. But somehow honey with a similar content of fructose to these is perceived as Archangel Michael. Allulose is not healthy. It is simply the other side of the homeostatic state of optimal health, free of exogenous carbohydrates. It is healthy in the context of mitigating cellular damage from an excessively inflammatory, glycating, fat-generating state of carbohydrate utilization. But remember the end result of a ketogenic diet. Once you lose the excess weight, your body weight does not change on a day-to-day to weekly to monthly basis. The concept of homeostasis. The body functions on the intrinsic determination of your genetics to maintain a seemingly optimal level of availability of cellular energetic substrate, namely fat stores and circulating glucose. You want to always have some fat on your body, otherwise you would starve after a day. You want to always have some circulating glucose in your blood, otherwise you would have no energy for certain parts of your brain and other dependent organs. Losing fat percentage at an unimpeded constant rate is not healthy. You would eventually reach zero fat stores, zero glucose from gluconeogenesis, zero cholesterol production, and you would promptly die from energy starvation. Your body wants a specific amount of fat stores at all times, not too much and definitely not too little. Your passive metabolic rate is constantly striving to reach equilibrium, the perfect balance between a minimum level of lipid energy reserves for a time of food lack, but not too much whereby it hinders physical performance due to the increase in weight. The homeostasis of glucose is meant to have a constant, stable circulating supply for your entire body to instantly utilize for glycolysis for energy production as needed. To bring it back to the sugar conversation, excess glucose and fructose, which is any amount over 0 nanograms, generates too much. Allulose makes you generate too little. The conversation around metabolic dysfunction is focused on hyperglycemia. However, hypoglycemia can also be pathological and result in dangerous states of being, such as low blood pressure, fatigue, arrhythmias, and palpitations, among other things. I think it is foolish to blindly trust a molecule that is literally fructose with one molecular position flipped on its head, and that's going to solve problems like it's some kind of philosopher's stone of metabolic health. However, I must admit that in anecdotal practice and clinical findings, it seems that in animal and human utilization, allulose does not significantly alter major glucosic or lipidic processes long term and is considered relatively safe for now. Alright, now that I've got my negativity narrated and got your noggin jogging, let me activate your oxalate containing almonds with some explanations of how it works. Again, I would like to emphasize that I am not a nutritionist, endocrinologist, metabolic expert, etc. I am simply giving my explanation of what I've read and watched. Hopefully it reaches a level of acceptable accuracy based on the current accepted understanding. I did the research because I wanted to, so that you don't have to. Alright, let's get explaining. You've maybe heard the term carbon-based lifeforms. This sentence is a critical mantra to comprehend biochemistry and molecular metabolism. All of existence is made of matter, of mass, of atoms and elements. Hydrogen, oxygen, carbon, nitrogen, phosphorus, and many others, these are elements, mass, that pervades the composition of your very body, and the numerous specific variations and combinations of these elements are tied to your structural makeup and your health. But the main concept that needs to be imprinted is that your body does not operate using calories or burning food, but that it takes food structures made of molecular mass, of atoms of carbon, of hydrogen, of oxygen, of nitrogen, and modulates them, metabolizes them, and chemically alters them into different molecular structures based off of the cellular and energetic needs that your health and hormones determine at any given moment, after which it will store, use, and excrete molecules. It is not calories in, calories out. It is molecules in, molecules out. Mass in, mass out. For a quick concept, the oxygen we inhale is a molecule of only 2 oxygen atoms. So, where did the carbon in the CO2 that we exhale come from? Well, it's from the food that we eat, and that food's metabolic changing in use for energy breaks down, and then you excrete the carbon after the job is done. Carbohydrate, the word itself means carboned water. Carbohydrate. What are glucose and fructose? They're C6H12O6. 6 carbon, 12 hydrogen, 6 oxygen. In the case of glucose, 5 molecules of water, 1 oxygen atom, and 1 aldehyde group, bonded together through carbon atoms in a hexagonal pyranose structure. Fructose has the same molecular composition, aka it's an isomer, but it has a furan structure of a pentagon, with a ketone functional group. Allulose is a C3 epimer of fructose. That means that molecularly, the hydroxyl group, the hydrogen-oxygen, at the third carbon linkage position, is flipped in atomic space. The relevance of being able to conceptualize these physical and chemical structural differences is key to not just understanding the differences between the 3 sugars, but to the greater concept of metabolism, absorption, and cellular utilization as a whole, among other things. Biochemical molecules that have sufficiently similar structure have the ability to utilize similar pathways, yet resulting in different outcomes, as I will explain soon. Because of this almost identical structure, allulose has been shown to be absorbed in the gastrointestinal tract through the same pathway as fructose, the GLUT5 and GLUT2 transporters in the luminal and basolateral sides of enterocytes. However, working sequentially, before being absorbed by the intestine, Allulose stimulates GLP-1 secretion, so let's get into that first. So, when a human ingests food, it is deposited into the stomach and subsequently transported into the duodenum and small intestines. This is where secretion of GLP-1 commences before their absorption by enterocytes into the bloodstream. There is a long cranial nerve called the vagus nerve that links your brain to your digestive system, among other functions. When the vagus nerve detects macronutrients being processed, it triggers the pancreas to release the relevant digestive enzymes into your digestive tract. In the case of carbohydrates, these include amylase, sucrase, and maltase, intended to break down starches and polysaccharides such as bread, fruit, cane sugar, corn syrup, and honey into simpler sugars such as glucose and fructose. This process is extremely rapid, exemplified by the flawed glycemic index, wherein some glucose from digested carbs can reach the bloodstream within minutes after ingestion. Relevantly, along with the digestive enzyme secretion is GLP-1. So, glucose, fructose, and allulose all stimulate GLP-1 release. Allulose is not unique in this. It is not nature's Ozempic, a term which is itself a fallacious comparison of the mechanisms of action of the GLP-1 hormone and Ozempic, a GLP-1 receptor agonist. So, the GLP-1 activation. GLP-1 is an incretin hormone released by the L cells of the small intestine. As an assumptive reminder, the hormones insulin and glucagon, regulators of blood glucose, are released by the pancreas by beta cells and alpha cells respectively, in response to increases and decreases in glucose. So remember that GLP-1 is released in your digestive tract in a similar manner, the detection of nutrients. From this paper, glucagon-like peptide 1 is an intestinally produced hormone secreted by the L cell. It is released in response to meal intake and plays an important role in glucose homeostasis. GLP-1 lowers postprandial glucose levels by potentiating glucose-stimulated insulin secretion and by inhibiting postprandial or post-meal glucose absorption rate through delaying gastric emptying, the emptying of your stomach into your small intestine. GLP-1's glucose-lowering effects are further amplified by a concurrent inhibition of glucagon secretion. But another counterpoint to the hype around Allulose's GLP-1 stimulating effects is that it is based upon a proposed fallacy. A common descriptor of allulose is that it is nature's Ozempic, which is simply a false equivalence. It is, in fact, the other way around. GLP-1 is already in nature. Ozempic is the unnatural pharmaceutical industry's GLP-1. Ozempic is a molecule called semaglutide that is actually a GLP-1 receptor agonist. By binding to the receptor proteins, it stimulates similar gastrointestinal effects as GLP-1. In other words, unnatural Ozempic mimics natural GLP-1. There is no such thing as nature's Ozempic, and there never was. Allulose and the other macronutrients, such as glucose, fructose, protein, and fat, are detected by pancreatic cells and stimulate the actual release of GLP-1. They are not GLP-1 receptor agonists. Let me emphasize that last point. All of the macronutrients release GLP-1. Fructose itself stimulates GLP-1. Allulose is not special in this, even among the sugars. It is a natural regulatory response to all food entering your body, allowing your digestive system time to process and break down food to prevent them from overloading your bloodstream, supplementing satiety signaling to prevent overeating food beyond your digestive capabilities. Glucose spikes GLP-1 much more than protein and fat. And why is this? Because your body absolutely does not want excess glucose in the blood. And then you ate carbs, aka unnecessary glucose, and so the body reduces its endogenous production of glucose and slowly tries its best to absorb the dietary glucose for processing in a rapid yet homeostatic method, the insulin spike, leading to superphysiological glycogen from excess glucose that is not needed for immediate cellular utilization. Then, when insulin is low, it's right back to gluconeogenesis. Your body does not run on dietary carbs. It runs on generating glucose from fat and situationally dependent levels of lactate. And when you eat carbs, your body instantly sequesters it out of the way and gets right back to doing what it was doing. Gluconeogenesis still occurs during glycogenolysis. The sentiment among carbohydrates is that they cause overeating, but this is still not unlimited. Although lower in comparison to protein and fat, you can still feel full from eating a carb-heavy meal. So, why is there such an emphasis on allulose in particular for this point? Well, it has to do with the comparative rate of absorption to the other macronutrients. Although gastric emptying is reduced, it is still ongoing, and assuming allulose was consumed along with other carbohydrates, such as starches which break down to glucose, and fructose-containing foods, eventually these saccharides will start competing for absorption into the bloodstream through intestinal enterocytes, which are cells in your intestines that facilitate absorption of food into your bloodstream and lymphatic system. This is where another of allulose's anti-glycemic effects come in. Enterocytes contain cellular glucose transporters, abbreviated as GLUTs, that allow nutrient transfer from the small intestines into the bloodstream. They are located on opposite ends of the cell, called the luminal and basal sides. On the luminal side for glucose, there is sodium-dependent glucose transporter 1, or SGLT1. For fructose and allulose, the main transporter is GLUT5. Glucose transporters are distinguished from each other due to distinct mechanistic action and affinity for nutrient uptake. For example, SGLT1 and by extension glucose requires sodium, while GLUT5 does not. This all ties into the concepts that the monosaccharides are not metabolized equally or even similarly throughout the body. Once inside the enterocyte, glucose, fructose, and allulose are all diffused into the bloodstream circulation through GLUT2. Note that certain glucose transporters can be located in various cells throughout the body with certain variants and specificity. The metabolic significance of the variety of transporters is that they are ubiquitous to the trillions of cells in your body, and the different transporters have different mechanistic actions of activation. For example, SGLT1 is sodium dependent, GLUT4 is insulin dependent. Getting a picture? The relevance towards allulose absorption is that in comparison to glucose and fructose, allulose transport through GLUT2 is markedly slower. Why is this? Well, mechanistically speculating, it could be the structural similarity allowing for its initial access, but the subtle structural difference leads to interference in its transport. A proposed example would be the flipped carbon grinding against the transporter walls, causing friction. But the thing to remember is that when consumed concurrently or consecutively, these three sugars will compete and cross-inhibit each other for absorption and transport. Glucose and fructose each have individual rates of transport, but when taken together, such as in sucrose, aka table sugar, or corn syrup, or honey, or fruit, they reduce each other's efflux into the bloodstream by about 50%. Allulose, when added to the mix, can increase this reduction to around 70%. It's like two lanes in a highway merging into one. The cars of glucose and fructose slow down and take turns filling in. Allulose can be seen as an 18-wheeler truck that you just know slows it all down more than usual. To bring it back to the GLP-1 conversation, GLP-1 levels are stimulated and sustained by nutrient detection in the intestines. So if allulose has a longer duration of digestion, then consecutively it will have a prolonged stimulation of GLP-1. Now, the thing about GLUT2 is that it has a significant role in glucose, fructose, and allulose cellular absorption. Unlike epithelial intestinal cells, which have a luminal and basolateral side containing corresponding GLUTs, the liver hepatocytes primarily use GLUT2 as a bidirectional transporter for glucose, fructose, and possibly allulose absorption and release from and to the blood. The pancreas also uses GLUT2 as a glucose detection transporter, stimulating insulin release. In normal metabolism, liver cells are perfectly adapted to use them as needed based on detected levels, with concentration gradient determined absorption and generation to maintain homeostasis. Mechanistically speculating, if a foreign molecule, such as allulose, is introduced and physically blocks these transporters, this inhibits their function and thus subsequently lowers circulating glucose absorption and insulin release. In addition, there is a rat study wherein allulose administration also augmented hepatic glucokinase translocation. Glucokinase is an enzyme that catalyzes the conversion of glucose into glucose 6-phosphate. So, concurrent to the lowering of gluconeogenesis, your liver will still uptake and additionally metabolize the reduced circulating glucose, and depending on your cell's cellular energy demands, utilize it for glycolysis in the TCA cycle or convert it into glycogen, further supplementing glucose sequestration. In conclusion, mechanistically speculatively speaking, allulose works by inhibiting and modulating your homeostatic mechanisms of cellular carbohydrate absorption and utilization, with the resulting lowered insulin levels, increasing rates of lipolysis or fat breakdown, possibly additionally mediated by the cellular transporters for glucose absorption being inhibited, resulting in preferential absorption of fatty acids into the cell through CD36 proteins for energy production, e.g., the Randle cycle, leading to metabolic processing in the Krebs cycle, resulting in increased rates in expulsion of excess unneeded or otherwise underutilized mass in the form of ketones. Notice how I did not mention calories. Now, although I focused on the relevant primary transport of glucose and fructose through GLUT2 and GLUT5 in liver and intestinal cells, there are numerous other GLUTs that actively utilize glucose throughout the body. One relevant example is GLUT4. Is it possible that allulose's effects could be additionally mediated through it? Mechanistically speculating, it depends on certain factors. For one, GLUT4 is insulin dependent, meaning cells that use GLUT4 for glucose uptake, such as muscle cells, require insulin as a preliminary signal for translocation of GLUT4 from the interior of the cell, aka the cytosol, to the cell membrane to allow glucose transport to the interior for metabolism and storage of glycogen. Now, quickly mechanistically speculating, but allulose, due to its downstream effects of having an insulin-reducing effect, would actually reduce GLUT4's effectiveness for cellular glucose uptake. However, that is not the full story, and I will get into that later in the video. But what I can do right now, in this moment, is ask you that if you're still following this so far and think that it makes sense, consider liking the video. Maybe subscribe. If you got questions or considerations or know where I went wrong, or if I go wrong further in the video, leave a comment down below, yo. Moving on to metabolism, allulose has been demonstrated to be poorly absorbed and utilized for enzymatic processing by cells, with a majority of it being excreted in urine unchanged. Why is this? Well, it ties into the relevant enzymatic processes of fructose metabolism. Much like glucose, fructose can be biochemically metabolized and generate the relevant substrates for cellular energy, such as undergoing phosphorylation and generating pyruvate in the process. Phosphorylation is catalyzed in the first step by the enzyme ketohexokinase, aka fructokinase. Bringing in this in vitro lab study of rat hepatocyte liver cells, the authors explain that fructokinase requires a furanose structure consisting of 5 carbon linkages to operate. Thus, the enzyme successfully processes several other fructose analogs. However, allulose was not found to be a substrate for fructokinase, which suggests fructokinase requires the hydroxyl group at carbon 3 trans to the CH2OH group at carbon 2. Carbon 3, as I mentioned earlier, is the linkage position that is flipped between fructose and allulose. All the other furanoses have the same structure there as fructose, giving validity to the hypothesis that the physical structure of the molecule is what is critical to the inefficient absorption and metabolism. And so, in theory, in vitro, allulose enzymatic catalysis is blocked nearly instantly in comparison to fructose, resulting in its lack of metabolism. This is a potential source of why allulose does not raise blood glucose, in contrast with fructose, which does so indirectly through epithelial and hepatic processing and conversion into glucose. Let's get into the AMP kinase activation. Okay, so long story short, the mitochondria is the powerhouse of the cell, or some other outdated simplified explanation like that. The cells of your body utilize adenosine triphosphate, aka ATP, in order to generate energy to produce cellular work. Basically, life and movement. Your metabolism consists of physical mass and oxidizing chemicals and exchanging electrons between them, not the intake of estimated heat units. Downstream of the ingestion and metabolism of energetic substrates such as fatty acids or glucose, enzymatic processes break these masses down into elemental molecules such as pyruvate, acetyl coenzyme A, and citrate that eventually result in ATP, carbon dioxide, and water, the latter of which are used and excreted in ways that you can guess. ATP stands for adenosine triphosphate. Adenosine is a nitrogenous nucleotide base that is attached to a carbohydrate named D-ribose. It's basically an organic chemical building block at the cellular level. Now, simply put, when this adenosine is phosphorylated or has elemental phosphorus attached to it, it becomes a relevant compound in cellular metabolism, ATP, adenosine triphosphate. Now, when this triphosphate is used for cellular energy, it becomes dephosphorylated, becoming ADP, adenosine diphosphate, then AMP, adenosine monophosphate. AMP can be thought of as a molecular signal that cellular energy levels have been depleted, and if you want to continue living, your cells need to generate more ATP or add more phosphorus to the AMP, because you need two more on the AMP to become ATP, which can generate cellular energy, and this process is facilitated by a sensory enzyme protein called AMP activated kinase, AMPK. So, how and why does allulose activate AMPK? Well, mechanistically speculating, a number of ways. Due to the inhibition of cellular utilization of glycolysis, possibly by its action of glucose transporter inhibition, there is less pyruvate that is catalyzed into acetyl coenzyme A, with downstream effects leading to a reduced production of glycolytically produced ATP. Subsequently, through concepts related to the Randle cycle, such as reduction of malonyl coenzyme A, this reduction in pyruvate results in cellular adaptation towards increased fatty acid oxidation pathways. Thus, AMPK is needed as a signaling molecule to stimulate lipolysis, or fat breakdown, with the resulting long-chain fatty acids sourced from your fat cells covering the synthesis of ATP. Long story short, when the cells utilize fatty acids instead of glucose, ATP synthesis is much more active and efficient. For example, the oxidation of palmitic acid, a 16-carbon saturated fat, produces 106 molecules of ATP. The oxidation of glucose only produces around 32. Yet another point why fatty meat and carnivore is a proper fuel system, and carbohydrate implemented keto, animal-based, and omnivorous diets are suboptimal in efficiency. So, when consuming allulose, you reduce the cellular utilization of glucose for energy, thus leading to an acute reduction in available ATP, signaling activation of AMPK to stimulate the production of more. However, since the cellular transporters for glucose are being inhibited, the cell goes through a metabolic shift towards an increase in lipolysis. But it's time to conceptualize this. Your cells use and store ATP in order to do energetic work. You think that stimulating AMPK through eating allulose is a good thing? No. AMPK is activated by low levels of ATP. When you eat allulose, you are reducing your cell supply of ATP. This is not a good thing. However, because your metabolism is robust and has adaptive processes in place of contingencies like this, you then switch to alternative generative pathways to maintain the redox status. So allulose is not improving your health. Your body is just adapting to a poison that is blocking the supply of energy. The enzyme AMPK has many, many functions. You cannot just hear AMPK and think, oh, it increases metabolic rate. Oh, I'm generating more ATP. There's a good explanation video by JJ Medicine on this, and I'll be tying in some of the points here. Okay, so it inhibits mTOR. This inhibits protein synthesis. mTOR's anabolic pathway is stimulated by elevated insulin and sufficient ATP levels, signaling cells to grow due to sufficient mass available. Remember, allulose lowers insulin and depletes ATP, so it's counterintuitive towards these necessary prerequisites. So if you are a bodybuilder on a bulk, allulose may be an inefficient substrate to utilize. However, if you're on that autophagy kick, AMPK also activates ULK1, which regulates autophagy. When ATP is low, the cell will recycle proteins in order to mitigate the stress brought about by the energy deficit. So you eat allulose and reduce ATP, your cells now have to recycle proteins to maintain themselves. With the ATGL, note the lipase. It facilitates fatty acid catabolism. So, allulose may increase fat oxidation. Emphasis on the oxidation, not burning. You don't burn fat. But anyway, this tracks with the clinical findings of increased plasma and free fatty acid levels, because fat is one glycerol molecule with 3 fatty acids attached to it. Thus, allulose's downstream effect of decreased glucose levels will coincide with increased lipolysis markers, which are on their way to cover the energy deficit. AMPK also inhibits acetyl coenzyme carboxylase, which inhibits fatty acid synthesis because of its downstream reduced production of malonyl coenzyme A, which then disinhibits CPT1, or carnitine palmitoyltransferase 1, allowing more fatty acids to enter mitochondria for beta oxidation, which is good because there are more fatty acids flowing throughout the blood. AMPK also phosphorylates TBC1 domain family member 1, and this protein facilitates the translocation of the insulin-dependent GLUT4 receptor to the cell membrane, and it does this independent of insulin signaling, thus increasing the corresponding cell's ability to uptake glucose even in a state of allulose-stimulated, GLP-1 stimulated, reduced glucose stimulated, reduced insulin, doing so through a different AKT protein phosphorylation pathway. And finally, the point where AMPK inhibits glycogen synthase. Glycogen synthase is an enzyme that catalyzes the reaction to produce glycogen. Allulose has been shown to increase glycogen levels in the liver. So how can it do this through AMPK's inhibitory action on glycogen synthase? Well, it's all back to the biochemical concept of molecular structure as well as signaling cascades based upon energetic demand. Glucose, when uptaken into cells, must first be phosphorylated into glucose 6-phosphate. The 6 means the phosphorus is attached to the 6th carbon linkage on the glucose molecule. This G6P can undergo numerous metabolic routes depending on cellular needs, such as the glycolysis pathway, to generate ATP if necessary if energy demands are immediate. However, if ATP levels are sufficient, then G6P can then be converted into glucose 1-phosphate, with the 1 being the first carbon linkage position. G1P is then converted to UDP glucose, and then glycogen synthase removes the UDP, and what is left is molecules of glucose attached to other molecules of glucose through glycosidic bonds, aka glycogen, which can be considered a nearby immediate source of glucose. Allulose potentiates glucose uptake in cells for immediate use, but if glucose is not necessary at that moment because ATP levels are sufficient, then it is converted into glycogen. The mass is the same, all that changes is where it is and when it is used. It's all still glucose. The phosphate molecule just changes from one corner to another, and the specific configuration undergoes specific intended metabolic reactions, much like fructose versus allulose. Now, in the liver, even though glucose uptake through GLUT2 is inhibited, it is still ongoing. Glucose transporters are capable of transporting multiple molecules simultaneously. Allulose does not completely inhibit it. However, remember the Randle cycle in that when cellular uptake of glucose is reduced, then long-chain fatty acid uptake is increased. With allulose-stimulating glucokinase, the reduced glucose undergoes increased rates of phosphorylation, thus ATP generation from fatty acid utilization and its capacity demands are soon met, because fatty acids generate nearly 4 times as much ATP as glucose. But glucose is still being metabolized at an increased rate, but is unnecessary for ATP generation, and so it is converted from G6P to G1P to glycogen for localized storage for a later date when the energy demand for ATP through glucose goes back to normal. Generating glycogen is not healthy. It's just consolidating unnecessary glucose nearby because the cell took it in while not needing it. As a quick final point, the thing about fructose is that it depletes ATP levels. So one is left wondering why it doesn't stimulate AMPK through the relevant regulatory mechanisms. Well, this is because fructose metabolism has a multitude of unique properties. The catalytic phosphorylation of fructose through ketohexokinase, aka fructokinase, is rapid, without feedback control in comparison to glucose. This swiftly depletes ATP levels faster than the cells can regenerate. In addition, there's this breakdown of subsequent AMP into uric acid, and uric acid is an inhibitory molecule towards AMPK stimulation. Thus, the rapid buildup of fructose-generated pyruvate leads to downstream effects resulting in fatty acid synthesis and reduced metabolic rate. So, to wrap up this story, the overarching theme of the mechanistic action of glucose, fructose, and allulose and your body's metabolism as a whole is time and energy, the rate of processing. Your body does not do things instantaneously. There are biochemical reactions that provoke subsequent reaction and effects, and these are dependent largely upon the chronological rate of how much mass is at any given time being processed and metabolized, and how much mass at any point in time is needed to generate energy. Glucose, fructose, and allulose and their effects on your cellular metabolism are mediated on their own enzymatic timescales. The overall moral of the story is one of eating specific masses of macronutrients and overeating beyond your chronological digestive and metabolic capabilities in a day, as well as simply eating the wrong things, such as carbohydrates like ultra-processed food, fruit, and honey, and non-digestible, energy-depleting monosaccharides. The ketogenic diet mantra is to keep carbs under 20 to 50 grams a day. Why? Because 50 grams of exogenous glucose is what your liver and endocrine system can reasonably process in a day through metabolizing and excreting that mass in order to maintain homeostasis. A day is 24 hours, so in a keto diet, it's not just the mass of carbohydrate you're considering, but also the time scale you intake that mass is equally as important. Allulose works by blocking transporters and giving you more time. It's an inhibitory stopgap against glucose and fructose absorption and contraindicated overloading of these energetic substrates because too much glucose and fructose glycates cells, it increases inflammation, it feeds cancer, it generates fat that immobilizes you, and many other illnesses. Allulose is not inherently healthy. It is just reducing the rate you're consuming and absorbing unhealthy food that causes these illnesses. What is actually healthy are macronutrients that are appropriate for human beings, such as animal protein and animal fat that contain all of the optimal levels of the optimally bioavailable macro and micronutrients, and excess intake of those are quickly and safely excreted in urine and feces and ketones. What is healthy is not a carbohydrate that isn't absorbed and metabolized and subsequently blocks absorption and metabolism of other energetic substrates. What is healthy is not a fallacious and nonsensical concept such as reducing hunger, thus reducing an intake of theoretical, inconsistently calculated calories. A ketogenic diet of 20 grams of carbs is considered healthy, but a carnivore diet of 0 grams is the most healthy. In that optimally healthy state, your body's homeostatic regulatory pathways of gluconeogenesis perpetually produces the optimal level of circulating glucose for energy needs. When you administer allulose, you inhibit pathways that mediate the generation and absorption of that circulating glucose. And does that make you healthier? No, and you'd realize this by the simple fact that during and after allulose intake, your body ramps up lipolysis or fat breakdown to generate more glucose and produces increased fatty acid energetic substrates to cover that acute deficit and attempt to reattain that previous level of optimal health in the cells. The body does not maintain that lower level of glucose. You always want some glucose in your blood, a minor surplus, if you will, because your brain and some other organs needs glucose every single second. Otherwise, you would die if it suddenly stops. The reduced glucose levels in the blood from allulose quickly goes right back to normal because the normal level is what your body determined to be minimally safe enough to cover any contingencies of energy needs. Reducing fat from allulose is not inherently healthy. You always want some fat on your body because, again, that's where the glucose comes from. And if all your fat goes away, you would starve and die. That's why in a sufficiently nutritious ketogenic diet, although you're unnecessarily focusing on burning fat, you generally do not drop below a certain ubiquitous body fat percentage of around 14%. Even when consuming excess carbs, aka extra glucose you didn't need, your body's circulating blood glucose is still at least partially generated by fat through gluconeogenesis. You know obesity is unhealthy, but do not forget that anorexia is also unhealthy. In conclusion, Increasing glucose levels in the blood with dietary glucose and fructose from vegetables and fruits and sugar, bad. Decreasing glucose levels in your blood with dietary allulose, also bad. Allulose is not healthy. All right, that's it for now. There are numerous other clinical trials and hypotheses, theories out there that I omitted and skipped over, but I intended to express some of the more closer to confirmed mechanistic speculations. In addition, I recognize that I myself may also be under misconceptions and may have gotten some things wrong, as others have done. I hope I got some or most of it right. And as a reminder, this is all theoretical. I am not a biochemist or expert metabolic scientist. I just read and interpret what they write. RX Sugar, I am not really opposed to what you're doing. It's a free country and people are free to choose what to eat and buy. Think of it this way. We used to think alcohol like wine and beer and smoking was healthy. Now we know they aren't. Yet, are people going to stop doing those? I'm just unsatisfied with the super supportive slant and the spiels of your scientific spokesman. I'm just making a Harvey Dent in the Harvard-educated mindset by looking at the other side of the coin. Thanks for watching. Like, subscribe, eat more meat. A lot more meat. Carbs in all forms are still unhealthy. But maybe I'm just like Saladino, just another idiot who misinterpreted science. Leave a comment down below, yo. And to perfectly close up this discussion, here's a succinct recommendation from the esteemed chef, Marco Pierre White.
Close your eyes and forget about calories for once in your life.